Trying to finish up most of the respiratory with you today. If we don't get all the way through it, then we'll finish it up on Thursday. I'd like to start the digestive system with you. And then, of course, we'll be gone for a week on break. When you come back from break, we'll try to work mostly on that digestive system that Tuesday that you come back. All right, so we've talked about our important dates before. So if you guys have lab tests on the 22nd and the 24th, does that mean that you want to do your lecture exam on the 29th?
Oi.
Okay. Everybody good with the 29th?
So we come back, it's going to be 24th, right?
So when you come back the Monday the 21st, and if y'all have lab tests that Tuesday and Thursday, then that kills that week.
Right.
Okay.
That's fine.
That test is chapters 20, 21, and 22. That means lymphatic, respiratory, and digestive will be on this test. The brunt of the questions come from 21 and 22. Those are longer chapters, there's more information in there, and there's definitely more physiology. So when you're splitting up your study time, make sure you spend more of your time on 21 and 22 and less on 20. Since you're going to be taking your test the day before the drop deadline, I like to have your grades for you that evening by 11 p.m. So that way you'll be able to make a decision about this class so you can take care of whatever you need to do on that Wednesday. That's going to be the 30th. Since y'all had mentioned this, I wanted to go ahead and pass the word along that Kevin is going to be here during break. He has volunteered to open up the lab for you all to study for your lab test Tuesday at 1 p.m. So 5410 is the place to be on that Tuesday if you want a little extra study time. Last class period, I'd also mentioned to you to take a look at the respiratory disorders. I believe these are on the bottom of page 80 in your course pack. And considering that we won't have the test until a good solid week after you come back from break, I wanted to give you at least a heads up to work on this during break, and then that Tuesday you come back, if you have any questions on these, we can do it then. Basically, I'm looking for the cause and or symptoms for these, and you might want to look at these as being possible essay questions. I don't yet have the test finished in terms of writing it up, but I can give you a better idea when you come back from break what to look for in terms of essay questions. But since I'm having you do this, I figure I might as well give you an essay on it because you've gone to the trouble of writing up the information anyway. So I'll at least make sure you have an essay question on this stuff.
Thanks.
Yeah, at least it makes you worth your while, right?
No. That was a sarcastic thing.
Thanks. But hey, at least you know what one essay is going to be.
That's true.
Are you going to do the A-B thing again?
Yeah. Is that good or bad?
Good.
Good, okay. Let's see, last class period we had gotten into the lungs. We had not enough time to talk about bronchioles and alveoli, so I'd really like to go back to page 74. Oh, sorry, not 74, 76. Before we start there, do y'all have any questions from last Thursday? All right. When we left off last Thursday, we had talked a little bit about the alveoli. What are alveoli? Good, the site of gas exchange in the lungs. How would you describe their structure?
Simple squamous.
Good. They're like bundles of grapes. They are very thin-walled. In fact, they're made of a simple squamous epithelial tissue. So they're only one cell layer thick. Some people describe them as being the thickness of a soap bubble. So that kind of drives home the point how incredibly thin-walled those alveolar air sacs are. They are clustered around the terminal endings of all those branched passageways in the lungs. You might recall that we started with the trachea being the windpipe, taking us down to the lower respiratory. The trachea branches into a left and right primary bronchus. It will then branch into a secondary bronchus, tertiary bronchus. What typically comes after the tertiary? Bronchioles. Yeah, after the tertiary, which is the third branching, we end up with bronchioles. And you'll notice here these bronchioles are surrounded with these little tiny clusters of alveolar air sacs. All right, so on your worksheet, let's go to the top of page 76. You'll notice that the alveolus is the functional unit of the lung. This is the whole purpose of having all these structures clean, humidify, filter all that air, because to get this air down to the alveolus, we want to make sure that what that gas exchange is doing is essentially working with clean, warm air. Alright, so you'll notice under the physiology there's a term there, it's called the respiratory zone structure. Part of the respiratory tree involves the bronchial tubes, which are no bigger than about 1 millimeter in diameter, and these alveolar air sacs. So gas exchange is taking place in this respiratory zone, which are these terminal branched endings called bronchioles, and those alveolar air sacs that surround them. Alright, so thinking back to Bio 210, in Bio 210 we talk about various membrane transport processes. Some are passive and some are active. How do you think blood gases move?
Passive diffusion.
Passive, yeah, by diffusion, good. So, kind of go back to Bio 210 for just a moment. How does diffusion work? Good. High concentration to low concentration. Any ATP required? No ATP required. And since we're talking simple squamous for those alveolar air sacs, this is going to be a real easy process for us to migrate those molecules from areas of high to low concentration. We also talked a little bit about surfactants in our last class period. You can see here, this is a real up-close view of an alveolar air sac. Here, for instance, is the lining of one. There are specialized cells that line those alveolar air sacs. They're called type 2 cells. Their job is to secrete that surfactant. Surfactant you might describe as an oily or lipid-type substance. What's the function of surfactant? Right, keep those air sacs from sticking together, because if those air sacs stick together, what can no longer happen?
Gas exchange.
Gas exchange, yeah. The lungs, of course, can't fill with air if those air sacs stick together. And if they can't fill with air, then we can't do gas exchange. We talked a little bit in last class about babies that are born prematurely. Surfactant's one of the last things to be developed in the body, during pregnancy before birth. So they can actually give the baby a synthetic type of surfactant to keep the air in the lungs, keeping those alveoli inflated until the body starts to secrete its own surfactant. But it really is necessary to prevent those lungs from collapsing in and sticking together. We also talked last class period about the types of structures lining the trachea, the primary, secondary, and tertiary bronchi that help to keep contamination and mucus out of the lungs. What lines the surface of all of those respiratory passageways?
Cilia.
Cilia, good. Those cilia are pushing contaminated mucus and debris up to your mouth, where either you cough it and get rid of it, or you can swallow it. And of course, the digestive tract is so acidic, it's going to destroy whatever you swallow that comes from the respiratory system. But by the time you get down here to the alveoli, no more cilia. So how do we now get rid of things that have made it past all this upper defense system? Macrophages, good. In your alveoli, there are no cilia, but we do have macrophages on patrol. They, of course, eat and destroy, so they're going to be removing any kind of bacteria or viruses or debris that make their way down here into these air sacs. All right. I want to go back and talk a little bit about the lungs themselves, because there's more to the lungs than just bronchial tubes, alveolar air sacs, and all these passageways. You'll notice that about halfway down page 76, we're going to talk about those lungs. The pointed portion of the lung is called the apex, and in this case, the pointed portion sticks upward toward the superior portion of the thoracic cavity. So you'll notice the apex of the lungs is both on the top up here. The base of the lung is the widest portion, and it actually rests down here on that diaphragm. So in terms of how the heart is situated, back when we studied the heart, the apex actually was the pointed portion of your ventricle, that oriented towards your left hip. Notice in this case, the apex actually sticks up there toward the top portion, up here toward the top portion of your thoracic cavity, that is. All right. In addition to those alveolar air sacs inside of the lungs, the lung tissue is also somewhat elastic. This is again thinking about what you learned back with the heart. If you have the stretch of the lungs as the lungs inflate with air, when the lungs deflate, what do you expect to happen?
Come back together.
Go back together. Go back to your flapping underwear concept, because if you have elasticity here that's gone bad, instead of recoiling back, what do the lungs do? Stay stretched. So in order to get the air up and out of your lungs, you're looking for elasticity that actually recoils back to the original shape, not stays inflated and stretched. So that's kind of a feature here of having our lungs operate properly, is for the elasticity in the lung tissue to recoil after inflation. Now, we have a little bit of help, because in addition to our lungs, we also have rib muscles, we have a diaphragm, and we also have muscles up here in the neck area that can help to push air out of our lungs. But the lungs themselves do need to recoil with elasticity after inflation. Part of this process is due to pleura, and I wanted to share with you the pleura, which are the membranes surrounding the lungs. Taking a trip back to Bio 210 again, you might recall we talked about a type of epithelial membrane called a serous membrane. These serous membranes are made up of 2 layers of simple squamous epithelium. Between those 2 layers, what is secreted? Serous fluid, good. What's the job of serous fluid? Reduce friction, good. So, let's take a look at this picture over here. You'll notice that we have a visceral pleura, which is actually the innermost. They got that labeled wrong, don't they? You notice that? It's backwards, isn't it? So whoever had this picture out there on the internet where I went looking for pictures got it backwards. The visceral pleura is the innermost of the 2 layers. It's like shrink wrap on the lungs themselves. The parietal pleura is the outermost of the 2 layers. If you want to say it's more of a loose sac, it is the external of the 2 layers. Here's something different. When we studied this on the heart, the outermost parietal pericardium on the heart wasn't really attached to your thoracic cavity wall. This time it's different. I'm going to get you a different picture because that one's bothering me. Let's go to another one. Let's do this one. Okay. That parietal pleura is actually adhered to your thoracic cavity wall on the inside. So if we were to have a needle, for instance, come into the thoracic cavity, after it passes through the muscles between the ribs, we would now encounter the parietal pleura. The parietal pleura is literally shrink wrapped to the thoracic cavity wall. Keep coming on in, you're now going to pass through that fluid layer between parietal and visceral pleura, and then now you're going to enter into what layer? Visceral pleura. I make this point because it's an important one for the way the lungs work. Think about when you have let's say you've got a cup of water or a cup of paper, something like that. If you sit it on a table and that glass sweats, when you come along to pick that glass up off the table, there's a little bit of suction, right? So you have to give a little bit of a pull and then the glass will separate. That's the case here because the fluid between the 2 serous membranes means that visceral and parietal layers stay stuck together. So if you inhale and parietal goes with the thoracic cavity, look who's coming along for the ride. Visceral. So visceral and parietal stay stuck together. Water is cohesive. Water's going to stick together. So that visceral and parietal layer is going to be moving in unison no matter how your thoracic cavity wall changes. So in the case of the lungs, we're looking at the parietal pleura actually being adhered to your thoracic cavity wall. So that means that if you breathe and inhale, all the lung tissue is coming along as well. Totally different concept from what we saw back with the heart.